Welcome to The Explainer. Okay, we are diving right into an absolutely incredible journey today. We're going to start with a squishy, raw biological tissue sample and follow its path all the way to a final digital spreadsheet, one that literally reveals the secrets of life. We're demystifying RNA sequencing. It's a truly revolutionary technology, and it's completely transformed modern biology as we know it.
To really get why this matters, we have to start with a foundational question. Think about it. If every single cell in your body contains the exact same DNA, why does a heart cell look and act completely differently than a skin cell? Well, as our sources explain, it all comes down to the central dogma of biology. See, DNA is static. It's the master blueprint locked safely away in a vault. But RNA is the active snapshot. DNA makes RNA, and then RNA makes the proteins that do all the actual heavy lifting in your body. So while your DNA blueprint is the exact same everywhere, the RNA tells us specifically which genes are actively turned on or turned off in one specific cell at one specific moment.
So here's our roadmap for The Explainer today. 1. The power of RNA. 2. Preparing the sample library. 3. Inside the sequencing machine. 4. Making sense of data. And finally, 5. Discovering differentially expressed genes.
Let's jump into Section 1, the power of RNA, the active snapshot of life.
Unlike DNA, RNA changes rapidly. It reacts constantly to its environment, to diseases, to drug treatments, or just to different developmental stages. And because it changes so fast, it gives us a real-time snapshot of what a cell is up to right now. We can actually look at this snapshot in two main ways, and the source documents use a brilliant analogy for this. Bulk RNA sequencing is kind of like a fruit smoothie. You take millions of cells, blend them all together, and measure the average gene expression. You know it's a smoothie, it tastes like fruit, but you can't pick out the exact source of every single flavor. Single-cell RNA sequencing, on the other hand, is like a fruit salad. You break the tissue down so carefully that each piece of fruit remains distinct. You can examine every single cell individually, which lets us uncover unique, rare cell types that would completely get lost in the blender. Okay, moving on to Section 2, preparing the sample library. This is our wet lab phase. This is the physical phase where our biological sample gets prepped for the high-tech sequencing machine. You can think of this whole process like managing an actual physical library of books. First, we have to extract the text. That's the RNA. Next, we create stable backups of that text. Then, we cut those really long books into shorter, readable chunks, or fragments, simply because the machine can only read short sentences at a time. And finally, we attach standard covers. These are called sequencing adapters, so that these fragments perfectly fit onto the sequencing machine shelves. Let's zoom in on that backup step for a second, because it's super important. Our sources emphasize that RNA is incredibly fragile. It's single-stranded and degrades just so easily. We literally can't put it directly into a sequencing machine. It wouldn't survive. So, scientists use an enzyme called reverse transcriptase to build a sturdy, double-stranded backup copy called complementary DNA or cDNA. From this point on in the process, we are actually working with this stable DNA copy, not the fragile RNA itself. Now, sequencing machines are pretty expensive to run, so efficiency is the name of the game here. Enter multiplexing. Just like stamping books to know exactly which branch library they belong to, we attach unique molecular barcodes or indices to the fragments of each sample. Because each sample has a unique barcode tag, we can physically mix dozens of different samples into one single tube. We run them all through the machine at the exact same time to save time and money, and we just let the computer sort them out later based on their barcodes. It's pretty genius. All right, Section 3, inside the sequencing machine. This is where we look at sequencing by synthesis. We are officially leaving the test tubes behind and stepping into the Illumina sequencer to witness what is basically a microscopic light show. Our barcoded DNA fragments get loaded onto what's called a flow cell. This is essentially a glass slide containing billions of tiny wells. Through a really clever chemical process called bridge amplification, single DNA fragments attach to the slide and are copied over and over again in a localized area until they form a dense cluster. I want you to imagine this glass slide transformed into a microscopic forest, where millions of identical DNA strands are just standing straight up, ready to be read. This setup perfectly illustrates sequencing by synthesis. The machine washes a solution of free-floating DNA bases, your A, C, T, and Gs, over our forest of strands. And here's the cool part. Each base has a different colored fluorescent tag attached to it. When a base naturally pairs up with our standing DNA strand, a laser fires.
A camera snaps a picture of the glowing dots, green for A, blue for C, yellow for G, and red for T. Then the chemical block is washed away and the whole cycle repeats. Flash by flash, color by color, the machine builds and records the exact sequence of millions of fragments all at the same time. Which brings us to Section 4, Making Sense of Data. Welcome to the Dry Lab.
So, what do millions of glowing dots actually mean for us? Well, this is where we pivot from the physical chemistry of the wet lab over to the digital software of the dry lab. We're entering the realm of bioinformatics. The sequencing machine actually translates all those colored light flashes into a massive digital text file called a FASTQ file. And when I say massive, I mean it. The sources note that a single run can generate a file with over 1 billion lines of data. This file stores the raw sequences, the exact ACT and Gs of every single fragment that was read.
But importantly, it doesn't just store the letters. It stores how confident the machine was when it read that specific letter, using something called a Phred quality score. Here's how it works. If a letter gets a score of 10, the machine is 90% sure it's correct. A score of 20 is 99% accuracy, and a score of 30 means it's 99.9% sure. This is absolutely vital, because our very first bioinformatics step is to look at these scores and automatically trim away or delete any of the low-quality garbage data where the camera might have been just a little bit unsure.
So, to go from these raw text files to actual usable biological insights, we follow a 3-step bioinformatics pipeline. First is quality control, which we just talked about, trimming the garbage and leftover adapters. Second is alignment, and third is quantification, which is basically just counting.
Alignment is fascinating. Remember, our FASTQ file just contains millions of tiny shredded sentence fragments. We have to figure out exactly where they came from. Using powerful software like STAR or HisSed2 that our sources mention, we map these tiny reads against a master reference genome. It is literally like solving a massive shredded jigsaw puzzle by looking at the picture on the front of the box. Once we figure out where every single piece belongs on the genome, we know exactly which gene it came from. And that leads us to our final step, Section 5, Discovering Differentially Expressed Genes. This is the payoff. To finally answer our original biological question about what is making these cells behave differently, we have to quantify our aligned reads, and that really just means counting them. We literally count how many puzzle pieces landed on gene A, how many landed on gene B, and so on. This turns our massive billion-line data files into a surprisingly simple spreadsheet called a count matrix. In this matrix, the rows are the genes, the columns are our biological samples, and the numbers are just the raw count of those fragments.
But hold on. Actually, scratch that. We can't just directly compare these raw numbers yet. We have to do some math called normalization. The sources are really clear that we must remove technical biases first. For example, think about gene length for a second. If gene A is physically twice as long as gene B, it's naturally going to get chopped into way more fragments during our library prep. So it might look like it's highly active in our counts, but it's really just a longer gene. Normalization mathematically adjusts for things like gene length and total sequencing depth, just to make sure we are making a fair apples-to-apples comparison.
Once everything is normalized, statistical tools step in to compare the counts across our different conditions, like comparing a normal tissue sample directly against a tumor sample. This process is called differential gene expression analysis, and it's where the biological magic finally happens. It tells us exactly which genes are upregulated, means turned on, and which are downregulated or turned off, driving a disease, a mutation, or a specific stage of development.
This reveals the ultimate payoff of our entire process. Imagine you're looking at gene A. In the normal sample, it shows almost zero expression. But in the tumor sample, the expression is completely off the charts. By tracing this entire journey, from a squishy tissue sample, through the wet lab chemistry, the sequencer's light show, and the bioinformatic puzzle, we have successfully isolated the exact biological instruction that is misfiring in this disease.
To sum the whole shebang up, RNA sequencing isn't just about reading a static code. It's about reading real-time activity. As we've seen throughout this explainer, RNA sequencing gives us a high-resolution snapshot of life as it's actually happening, opening up this incredibly dynamic window into the very machinery of our cells.
So, I want to leave you with this final thought. Now that you understand how we have the power to read the active, real-time instructions of absolutely any cell, what disease or what deep biological mystery would you want to decode first? Thanks so much for joining me on this incredible journey today. Keep asking questions and keep learning.